Have you ever thought about how biologists deal with our genes? It's like trying to read every single book in a massive library all at once. It's an insane challenge. But what if you had a tool that could instantly light up every book you were interested in, giving you a complete snapshot of the entire library? Well, that's pretty much the power of microfluidic technology. And today, we're going to break down how these tiny little labs on a chip are completely changing the game.
So, how are we going to unpack all this? First, we'll dive into the revolution itself, what this is all about. Then, we'll look at how these chips are actually built, and just as important, how we make sense of all the data they produce. After that, we'll explore their real-world impact, and then, of course, take a peek into the future.
All right, so to really get why microarrays are such a big deal, we have to start with the huge problem they were invented to solve.
I mean, really think about that for a second. The human genome has something like 20,000 genes. So how can a researcher figure out which ones are switched on in a cancer cell versus a healthy one without, spending their entire life on it? This was the central challenge. The scale of biology is just staggering.
And this is the brilliant solution. A microarray is basically a tiny, solid surface, usually no bigger than a microscope slide, that's covered in thousands of microscopic probes. When you add a biological sample, say from a patient, the parts of that sample that match the probes stick to them. And with the help of some fluorescent tags, they light up. Each one of those glowing spots tells you something specific is going on. A gene is active, a protein is there. It's a massive parallel experiment on one tiny little chip. And this tech hasn't stood still, not even close. It has evolved at lightning speed. It started in academia back in the 90s with people making their own custom arrays. Then in the 2000s, commercial platforms emerged and made them way more accessible. By the 2010s, the density of these things just exploded, which created a massive data boom. And today, well, we're pushing into incredible miniaturization and even using AI to make sense of it all. Okay, so how are these powerful little tools actually made? The real magic is in the details of how they're constructed. Let's get into the nuts and bolts.
So it's really important to know that not all microarrays are the same. The probes, those little bits of DNA that act like bait, they come in different lengths. Early arrays use longer, more variable probes, but a lot of modern commercial arrays use shorter, more standardized ones. Now, why does that matter? Well, shorter probes that are synthesized right on the chip let you pack an incredible number of them in, which means better reproducibility. The trade-off is they might be a little less sensitive. It all depends on the question you're trying to answer.
The physical format also changes things up. You've got your classic microarray, which is a solid, flat surface like a glass slide. This format is fantastic for sensitivity. But there's another approach: suspension arrays. These attach the probes to microscopic beads floating in a liquid. This lets you run a huge number of tests in a single tube, giving you incredible throughput and flexibility. It's that classic trade-off, right? Sensitivity versus sheer scale.
And get this, it's not just about what's on the slide, but what the slide itself is made of. Glass is standard, sure, but researchers are now using some pretty incredible new materials. Things like silicon and ceramic can actually enhance the fluorescent signal, making it easier to see what's going on. Carbon-based surfaces offer better stability. And some are even using nanostructured surfaces with tiny posts that amplify the signal, letting scientists detect even the faintest biological whispers.
Okay, so we have our chip. It's covered in all these glowing dots. Fantastic. It looks cool, but a picture of glowing dots isn't a scientific discovery, right? The next step is arguably the most critical part of the whole process: turning that raw data into actual biological insight.
Now, because this is a mature technology, you've got major industry players driving it forward. Companies like Affymetrix, Agilent, and Illumina, they each have their own unique platform. They use different probe types, different data formats, different everything. Now, this diversity is great, but it also creates a major headache, trying to make sure data from one platform can be compared to data from another.
To get from that raw signal to a real conclusion, the data has to go through a pretty rigorous pipeline. First, a scanner collects the raw intensity of every glowing spot. Then comes preprocessing and normalization. This is a super crucial cleanup step to get rid of background noise and fix any technical glitches. Only then can you start to apply statistical tests to find the differences that actually mean something. And the final step, of course, is translating those stats back into biology. What does this pattern of gene activity actually mean?
And this quote from our source material really just hits the nail on the head. The analysis is not one size fits all. The methods you use are deeply tied to the specific platform that made the data.
It takes sophisticated statistics and more, machine learning to pull that true biological signal out from all the noise.
All right, now that we've got the tech down, let's zoom back out. Why should we care about any of this? Well, because this technology is having a huge impact on medicine and science right now.
Yeah, microarrays are not just some obscure research tool. They are absolute workhorses in so many fields. In clinical diagnostics, they're used to spot things like chromosomal abnormalities. In genomics research, they're completely essential for understanding what genes are doing on a massive scale. They're also speeding up drug discovery and can even be used for things like environmental monitoring, like seeing what kind of microbes are in a water sample.
But here's where it gets really exciting. These tiny labs are actually leaving the lab. Thanks to miniaturization, we're seeing the rise of what's called point-of-care diagnostics. Imagine a device, maybe even hooked up to a smartphone, that can run a complex microarray test right at a patient's bedside or in a remote clinic. This isn't sci-fi, this is happening now, and it's a total game changer for getting test results fast.
But let's be real, it's not a perfect technology. Not yet. There are still some pretty significant hurdles to overcome. Integrating data from different platforms is still a massive challenge. Fully automating how samples are handled is tough. And maybe most importantly, taking a cool finding from a research array and turning it into a reliable, clinically validated test that doctors can trust, well, that's a long and difficult process.
So with all those challenges in mind, where is this whole field going? The future is all about making these tools smarter, smaller, and more integrated than we've ever seen before.
To get over these hurdles, the research community is firing on all cylinders. They're pushing hard for standardization so data can be compared more easily. They're developing fully automated lab-on-a-chip devices to cut down on human error. AI is being deployed for smarter quality control. And those new nanoscale materials we talked about are pushing the limits of sensitivity even further. And all of these advancements are really pointing towards one incredible goal: a fully automated, portable device that can analyze not just genes, but also proteins and other molecules all at the same time. A true multi-omics tool. The dream here is to finally bridge that gap between cutting-edge research and everyday clinical practice.
So this leaves us with one final thought. As these incredibly powerful diagnostic tools move out of the lab and maybe even into our homes, as they get smaller, faster, and smarter, how is that going to change your relationship with your own health information? That's something to think about.